Hello and welcome to the episode number 11th for the Hydescience Lab series. So today we are diving into acids and bases, specifically in some basic indicators and how do we determine if something is acidic or basic or alkaline, and how do we obtain quantitative and qualitative data about them. So we're kind of laying the foundation, gaining some background knowledge that we will need moving into some labs in the future, such as titrations. So this is kind of what our sheet looks like today. We have several data tables to fill out. Now this video will not dive into the last two sections of this. There are some vlogs posts that I have put up in the past that really dive into detail about those if you want to check those out. And that'll be the part on the buffers. So let's get started. All right, so this is kind of jumping to the end results that we will get kind of used to fill out our data tables. So we're using three indicators. One is blue litmus paper, we have red litmus paper, and we have pH hydrion paper. And what we do is we will dip them in our substances. We have some pretty common chemicals. We have Windex, alcohol. This is isopropyl alcohol, pickle juice, Tums, sugar solution, Coca-Cola, sodium hydroxide, baking soda, water, lemon juice, salt solution, bleach, and hydrochloric acid, which we call stomach acid. And this just kind of matches our diagram here on our handout so we can keep up with everything. How we test this is we will take the strips. So we have up first our blue litmus paper, and we dip that into our solution, and then we can lay it out. Then we will take our red litmus paper, do the same process, and lay it out. So we will analyze these two first, as they kind of work complementary to each other. So up here is Windex. It's kind of been sitting for a minute, and we can see that the red litmus paper has turned blue. So this indicates to us that it is a base. And the blue litmus paper stayed its original color of blue. So we have like a double blue result here. So it indicates it's a base. You might be thinking, why don't we just use one of these colored litmus papers? Because it may not be enough. If we jump over here to our sugar solution, we see that the red litmus paper stayed red, the blue litmus paper stayed blue. So that indicates to us that it is neutral. Had we have only used the red litmus paper, we would see, okay, well, it didn't change, so it must be a base. Or if we use the blue litmus paper, we'd say, okay, it didn't change, so it must be acid. So we need those to complement each other. And this is qualitative data. So these are good at giving us qualitative data, and that's just simply, is it an acid, is it a base, is it a neutral? So no data, no numerical data there. Up next, we will test the hydrion paper, and this gives us much more of a range. It does more than just turn red or blue. So again, we dip that into our solution, we lay it out, and then we can use our color code to match what we have. So we'll see that this matches, to me, the 8 fairly well. You see the color of the strip and the color of the indicator. So we know that this Windex is basic. It is alkaline. And we know more than just is it an acid or is it a base. We know the pH value roughly. So it's a pH value of 8. So this gives us some quantitative data. It gives us a quantity, a numerical data to record. So we run through all the tests, we record our results, and then we discuss bleach. So bleach is a base, and we see here that it stained or it bleached the paper. So in this case, these indicator methods are not the best tools to use. So if we were doing this and we really needed to determine information about bleach, we would need to turn to another indicator, and there are many indicators. If we had to make a conclusion based on what we see, we cannot identify any quantitative data because there is no white on our hydrion paper. But we can see at the top of the red litmus paper here, it did turn blue. So if we had to make an assumption based off of our data here, we would make the assumption that it is a base, or it is alkaline. So that wraps up this part of our acid-base analysis, the use of indicators, gathering quantitative and qualitative data. And that is it for this episode of the Lab series. We will see y'all with the next episode.